No, it's my pleasure to introduce Reece. He's a young man who's going to become a junior. He's going to start his junior year at Vanderbilt University in Nashville. So you can understand and appreciate how exciting it is to work in my lab, because I drug a kid from Nashville back to Springfield for the summer who's 20 years old. He reminds me a lot of my own son. He's 20 years old. He's got about the same haircut. He's inquisitive. He's got a lot of good ideas. He's got a bright future, which is shocking for me to say, because Tiffany and I are still 29 again. And so to have a 20-year-old who's about the same age as my own is exciting and interesting to have in the lab. So I'm going to let him tell you his story, but it's been my great pleasure. I think his parents should be proud of him. They've done a wonderful job raising this young man, and I wish him the very best in the future. I do hope, and I've tried to bring him back so that I hope that he does, if he decides to go to graduate school, that he'd come back and work with us. I didn't do a very good job because, shocking to all of us, everything worked except for one thing that they did this summer. So he's got a lot of exciting data to present, and I'll give the floor to him. Thank you very much.
So my presentation is called Characterization of Selective Chemical Inhibitors of TGF-beta Signaling Using a Quantifiable Cellular Assay. So first, we're going to talk about TGF-beta function in healthy physiology. So TGF-beta is a cytokine that is found everywhere in the human body, and it has a lot of different functions. And it binds to two different receptors called TGF-beta receptor 1 and 2, which phosphorylates SMAD2/3, which is then translocated to the nucleus, which is allowed to undergo a lot of different TGF-beta dependent gene expression. And now we're going to talk about the phenotypic and functional diversity of human natural killer cells. So in the bloodstream, they're called peripheral natural killer cells, and they have CD56 and CD16 receptors. And their main job is to kill cells infected by bacteria and viruses, and also cancer cells. These cells are also found in early pregnancy under the presence of TGF-beta, and they're called decidual natural killer cells, and they only have CD56 receptors. And instead of killing, they promote angiogenesis and promote healthy pregnancy. Similarly, in renal cancer, this process is very similar, as TGF-beta is also present, and the only receptor is CD56, and they also function in angiogenesis and do not kill the cells. So our main hypothesis is TGF-beta mediated inhibition of human natural killer cell function is preventable. This is that same process of phosphorylation of SMAD2/3, which translocates to the nucleus, and this makes the cells poorly cytotoxic compared to when there's no TGF-beta present, which makes them potently cytotoxic. So here's our cell-based assay to quantify TGF-beta signaling inhibition. This process occurs, it activates the SMAD promoter in the gene, which activates the firefly luciferase gene, which is the same gene found in fireflies that makes them produce light. And this produces light, which we can use to quantify TGF-beta inhibition. So our first experiment that we did was determining that TGF-beta response is time-dependent. So we used TGF-beta responsive cells and added TGF-beta in increments of 2 nanograms per milliliter at 2 hours, 4 hours, 6 hours, 8 hours, and 24 hours. And we use that to quantify luciferase activity. So our data from that shows an increase in luciferase activity as exposure time increases. There's a large gap between the 2 hour and 4 hour mark, and then it kind of levels off. So we used 3 hours as our exposure time for our next experiment for TGF-beta. Now we use 3 inhibitors to inhibit TGF-beta. The first one is called ITU1, and it inhibits TGF-beta receptor 1 and receptor 2. LY, which inhibits R1 at a low dose and R2 at a high dose, and SD208, which inhibits R1. And then we also compared their IC50 values. These are predetermined values. They're basically how much of the inhibitor causes 50% inhibition. So for ITD1, it's 3 micromolar. LY for R1 is 38 nanomolar. R2 is 300 nanomolar. And then for ST208 is 48 nanomolar. And ITD1 is much greater than the other two since it's micromolar compared to nanomolar doses. So here's our experimental design to assess inhibitor activity. So again, we use TGF-beta responsive cells. We had a control plate that was untreated with no inhibitor or TGF-beta. We had another plate without inhibitor or TGF-beta, and then a third plate with inhibitor and TGF-beta, and then we quantified luciferase activity to compare them. For ITD1, we found that it is a potent inhibitor of TGF-beta signaling, but is toxic at a high dose. As the dose of ITD1 increases, luciferase activity decreases. And if you can see the white bars, which represent no TGF-beta, around the 30 and 100 micromolar mark, they're much less than just the baseline values, which indicates that they're toxic to cells, which is not ideal. We found that the optimal dose of ITD1 for this is about 10 micromolar. For LY, we found that it is a potent inhibitor of TGF-beta signaling without the toxicity, so the baseline white bars are around the same throughout, so it's at high doses, they're not toxic, which is good. And with TGF-beta, it also decreases as well. And we found the optimal dose to be around 300 nanomolar. The results for ST208 are very similar. It is also a potent inhibitor without toxicity. The white bars are very consistent. They don't decrease past the normal value at high doses of inhibitor. And also we found the ideal dose of ST208 to be around 300 nanomolar. So to summarize, for ICD1, we found about 90% inhibition at 10 micromolar, which is slightly more than the IC50 dose of 3 micromolar. For LY, we found 93% inhibition at 300 nanomolar compared to the R2 value determined, which is also 300 nanomolar, so the same. And the ST208, which has 90% inhibition at 300 nanomolar, which is greater than the IC50 dose of 48 nanomolar. So for ICD1, we determined that micromolar dose is needed, and they're toxic above its IC50 value, which is not very ideal. And for LY and ST208, the results are very similar. They both require a nanomolar dose, and they're both non-toxic to cells. So either LY or ST208 are the most viable candidates for TGF-beta inhibition compared to ICD1. So for our future direction, we asked the question, can selective inhibition of TGF-beta maintain anti-tumor function of human natural killer cells? So if we introduce inhibitor to a tumor environment, can it inhibit TGF-beta so that the natural killer cells return to their cytotoxic form and can kill the tumor rather than support it? Thank you.
Any questions for Reece?
Yeah. I don't know, this question kind of can go to, I guess, any of the students, but do you guys compare, when treating cells, do you guys ever compare things like red light therapy or other holistic, non-invasive techniques?
We really did it for mine, but yeah, not really.
Dr. Wilbur mentioned that everything worked except one thing. What was the one thing that didn't work?
It was ITD1 not being as good of an inhibitor as we thought.
So what tumors are overexpressing TGF-beta such that you might be able to target?
So the main ones, I think it's on the first slide. We looked at renal cancer, so kidneys, I think.
Is that the only one? Renal cell carcinoma?
That's mainly the one we looked at, mainly, but.
Yeah. Almost all of them.
Almost all.
Yeah.
I thought I saw another question. Yes.
Yeah, I thought this is just way cool, very interesting, because NK cells are not class 1 HLA restricted, which is way cool, because they have the ability to recognize things and kill them regardless of their HLA. But now here we find something that's not letting them do what they do. This is very interesting. It would be interesting to see what their ability